Okay, so let's move on to section 7 of chapter 3, where we're going to talk a little bit about the organelles that can be found within a cell. Not all cells have all of these organelles, but these are the possible organelles that cells can have. Pretty much we classify the organelles based on whether or not they have a membrane. So we have some that have a membrane and some that do not. And where these organelles are found are within the area referred to as the cytoplasm, which consists of the cytosol, which is just the fluid-like substance that's within the plasma membrane, the organelles, whether they contain membranes or not, and inclusions, which are just little vesicles that can contain things. For instance, they could have glycogen granules in them or even some pigments. Okay, so let's start with our first organelle, which is the mitochondria. Your predominant focus when we hit these organelles and on the worksheet is really just to understand what the general function of these different organelles are. So the mitochondria can also be referred to as the power plant of the cell. Why? Because they're the ones that produce the energy. So they undergo, or it is the site where cellular respiration takes place. Cellular respiration is an aerobic process, which means that it requires oxygen in order for it to produce the energy, which is in the form of ATP. We'll talk a lot more about this when we hit the skeletal muscle system and the role of cellular respiration. It's not only bound or enclosed by a single membrane, it actually has two membranes right here. The outer membrane is pretty smooth, and then the inner membrane is going to go in and make some infoldings right there. You don't really need to worry about the cristae or the membrane proteins. However, I would be aware that the mitochondria is another site within the cell that does have DNA, as well as RNA and some ribosomes. So we typically think of the location of DNA as being solely exclusive function of the nucleus. However, you can find some DNA, which is called mitochondrial DNA, within the mitochondria itself. And it's actually going to be responsible for directing the synthesis of about 1% of the proteins that are required for the mitochondria to function. The other 99% would be encoded by the DNA that's found within the nucleus. Our second organelle is a ribosome. It's made up of something called ribosomal RNA or rRNA. So ribosomes are not proteins because they're not made up of amino acids. They're made up of rRNA. And they're basically the site where protein synthesis is going to occur. And the process of protein synthesis is called translation. And we'll talk about that more at the end of this chapter. So ribosomes can either be found freely floating in the cytosol, or they could be found bound to a membrane. And if they're bound to a membrane, the membrane is going to be called the ER, which stands for endoplasmic reticulum. And when they stud the endoplasmic reticulum, we say that the endoplasmic reticulum is rough. We also have an endoplasmic reticulum that does not have ribosomes bound to it, and we call it smooth because it appears smooth under a microscope. So a little bit more about the endoplasmic reticulum. Because the rough endoplasmic reticulum has those ribosomes that are studding it, that makes it appear rough, and because ribosomes are the site of protein synthesis, we would expect the rough ER to be involved in the synthesis of proteins. However, they're typically the site of synthesis of proteins that are going to be secreted from the cell, so not the proteins that are actually going to be used in the cell. It's also the site of a lot of plasma membrane proteins, the ones that are going to be embedded in the plasma membrane, as well as phospholipids. The smooth ER is a little bit different. It's not involved in protein synthesis because there are not any ribosomes associated with it. Instead, it's responsible for several different things. So one would be lipid metabolism, also synthesis of things like cholesterol and phospholipids, absorption and synthesis and transport of fats, detoxification of certain drugs and pesticides and even some cancer-causing chemicals, It's also able to convert glycogen into free glucose, and it's able to store and release calcium ions, at least in most types of cells like skeletal and cardiac muscle cells. So we would expect something because it's involved in detoxification, which is also one of the roles of the liver. So if we're looking at a cell called a hepatocyte that's associated with the liver, and we look inside at the organelles, we would see that liver cells have a huge proportion of smooth endoplasmic reticulum because they are the site of detoxification. Our next organelle is going to be the Golgi apparatus, and you don't really need to worry about the 3 steps involved. Just again, kind of have a general understanding of what the Golgi is responsible for, and pretty much it acts like a post office. So certain things like proteins and lipids that are being sent from the rough ER to the Golgi. The Golgi will act kind of like a post office where these proteins and lipids come in. You basically put a stamp on it, you put an address on it, and you ship it away. So the Golgi is responsible for basically modifying, tagging, like basically putting a location to where it needs to go, organizing it, and then shipping it off. Our next organelle is peroxisomes. The whole function of a peroxisome is really to detoxify a lot of substances and neutralizing free radicals. So they contain 2 important enzymes. One is oxidase, which converts the free radical into hydrogen peroxide. And then a second enzyme, which is called catalase, is going to take that hydrogen peroxide and convert it into water. So both of these enzymes are important. So if you have free radicals, those are extremely toxic to a cell, oxidase can go in and convert it into hydrogen peroxide, which is less toxic than a free radical, but still somewhat toxic. So then we need catalase to come in and convert the hydrogen peroxide into water, which is not harmful to our cells at all. Another organelle is the lysosome. We kind of looked at this a little bit before when we were talking about phagocytosis and bringing in either foreign microbes or cellular debris or anything like that. So that's pretty much their function. Is basically when you phagocytize something, that vesicle that was bringing in the foreign object is going to fuse with the lysosome, because the lysosome contains a ton of digestive enzymes that can go in there and break down and degrade bacteria, viruses, and various toxins, and even worn out and non-functional organelles in cells.